Hello, my name is Hussein Yassin. I'm an associate professor of medicine and neurology at Keck School of Medicine of USC. I'm an endocrinologist by training, and my lab is focused on understanding brain lipids, specifically in Alzheimer's disease. We focus on ApoE4. ApoE4 is the strongest genetic risk factor of Alzheimer's disease. Up to 25% of the population carry one copy of the ApoE4 allele. And up to 50% of patients with Alzheimer's disease have ApoE4.
Since ApoE carry lipids, our lab focuses on the relationship between ApoE4 and lipid, and it combines a multidisciplinary approach from basic science to clinical trials.
As you probably know, we have no effective treatments that can modify or slow down the progression to Alzheimer's disease. We need a new understanding of the key elements that are driving pathology and key targets that could be investigated to stop this disease and its effect on our aging population.
Our recent research identifies brain inflammation as a key target for drug development. We are working with colleagues at USC and internationally to identify drugs that can inhibit brain inflammation in the setting of ApoE4 dementia.
So inflammation increases the risk of cognitive decline and dementia in the brain. And ApoE4 is associated with greater inflammation in the brain. Our research has looked at that by examining the mechanisms that allow omega-3s and fatty acids to exchange in the brain. Throughout a series of experiments we have been doing over the past 3 years, Xiaowei Wang, a postdoc in my lab, identified that a specific enzyme known as cPLA2, or calcium-dependent phospholipase A2, is increased in ApoE4 mouse models. This finding was later confirmed in a cohort of human brains that differ by ApoE4 genotype. Then we collaborated with Seva Catrich at Michelson's, screening 18 billion small molecules, and landed with 3 potential candidates that effectively can block cPLA2, verifying that information in vitro in the lab and using cell models. To be able to identify which patients might benefit from such an intervention, we developed an 18F arachidonic acid PET probe. As you can see in this figure, 18F arachidonic acid uptake in the brain is shown in red in ApoE4 targeted mice and correlates with brain inflammation. We believe this would be a useful companion to identify patients with brain inflammation that could benefit from cPLA2 inhibition. What this paper tells us is that the activation of cPLA2 increases the liberation of arachidonic acid, which is converted inside the cell into inflammatory mediators. Since cPLA2 liberates arachidonic acids from membranes, it drives neuroinflammation. To us, that means that cPLA2 could be a potential target. And if we can block it or reduce its expression, we can offset neuroinflammation. The cPLA2 association with ApoE4 is novel, but it has been shown before that strokes, injuries to the spinal cord or to the brain, such as traumatic brain injury, activate cPLA2 and associate with greater inflammatory signaling response. In this case, we identified a subtle activation of cPLA2 with ApoE4. And chronic, subtle, repetitive activation of cPLA2 over a long time can lead to a powerful inflammatory and oxidative stress response that cascades into disease. So what are the next steps? Well, we're seeking collaborations and funding to make a drug that can inhibit brain inflammation based on cPLA2 activation. We need to synthesize analogs that can cross into the brain effectively. We need to determine the PK/PD safety profile of these potential medications. And finally, we need to translate the 18F arachidonic acid PET signaling into the clinic. We are excited to share this progress, and you can email me at hyassin@usc.edu or visit our lab website, yassinlab.com. Thank you.